The second portion of our Chapter 1 lecture deals with the scientific method. Scientific method is a topic that many of you are somewhat familiar with at this time, maybe all of you, and you realize that it's a particular series of steps that are used to gain information about the natural world in a non-biased manner. And so the scientific method involves 4 basic steps. The first step is to make an observation about the natural world. The second step is to propose ideas that explain that observation that you have noted. The third step is to form a test, to test those ideas that you have formed, the ideas that we call hypotheses. And the fourth step is perhaps the most important, and that is this, that you either retain or discard those proposed explanations based on the results of the data that are obtained from the test. And so that's the really important thing about the scientific method, and that really is what science is. Science should be based on data and nothing else. And so that fourth step tells us that if the data supports our hypothesis, we retain it and move forward. If the data does not support the hypothesis, then if we're ethical scientists, we reject the hypothesis and move on to another hypothesis. So what is a hypothesis then? A hypothesis is some explanation that can make sense of what we observe occurring in nature. And so there are scientific hypotheses and non-scientific hypotheses. In order for a hypothesis to be scientific, it has to be something that we can actually test. And so this means it can't be something that's attributed to the supernatural. It has to be something we can get our hands in and get our hands on and actually form a test to either prove it correct or disprove it. And then that's the last point we have on this slide. A hypothesis has to be falsifiable. You have to be able to prove that it's not correct, at least potentially prove it's not correct. That has to be an option as you test the hypothesis. We move on to the term scientific theory at this point. And so what is a theory? A scientific theory is a hypothesis that has been proven correct over and over and over again. And so remember that in our day-to-day life, we may use the term theory in a completely different manner. We may say, well, I have a theory that it only rains if you carry an umbrella or something like that. So some sort of kind of whimsical explanation. But in science, we reserve the term theory for a hypothesis that is well proven, super well proven over long periods of time. So one example would be the germ theory of disease. Fascinatingly, 200 years ago, we had no idea that bacteria existed and that they could be transferred from one source to another and cause disease. Today, we, of course, universally accept that that is the case, and that's referred to as the germ theory of disease, the idea that microorganisms, such as bacteria, viruses, fungi, can cause disease. So keep that in mind as we move forward. A scientific theory is a particular hypothesis that has been proven over and over again and that we have a great deal of confidence in at this particular point. So, is it possible that a scientific theory can be disproven? And the answer is yes, it can. And so we have a fascinating example of that on this particular slide. So Dr. Robert Warren and Dr. Barry Marshall won the 2005 Nobel Prize in Medicine. And so remember, we'll pause here for just a moment and point out that a Nobel Prize in every category except possibly the Peace Prize, which some people would argue is can be strongly politically motivated, is supposed to be the best, an award that goes to the invention each year or the discovery each year that has the greatest impact on humanity. And it's given out in all these different categories: medicine, biochemistry, chemistry, and it typically takes a lifetime to get a Nobel Prize. And so you come up with a discovery, and for years and years and years it has an impact on humanity, and then eventually, much later in life, if the effect has been significant enough, then you receive a Nobel Prize. It's not given posthumously as well, so it's got to be something that's significant enough to alter the world within your lifetime. And so it comes with a significant monetary award and a tremendous amount of prestige. And so these two doctors and researchers received this award for their work demonstrating that human ulcers are not caused primarily by eating spicy foods, but they're caused by a bacteria, Helicobacter pylori. Fascinatingly, for long periods of time, people believed that spicy foods were the primary cause of human ulcers. And these doctors, through a tremendous amount of research, much of it clinical and simply drawing correlations, much of the research focused on drawing correlations between people who were infected with this bacteria and people who developed ulcers. They proved that a tremendous percentage of ulcers are caused by far the majority of human ulcers are caused by this bacteria, Helicobacter pylori. Fascinatingly, they finished it up in a very dramatic fashion. One of the researchers did not have ulcers, and he drank a cupful of Helicobacter pylori, and you can guess what happened to him. He developed ulcers. Now, of course, that wasn't part of the official research, but that was a dramatic conclusion to the years and years of research drawing these conclusions. So the point of bringing this up is that can a scientific theory, and again, remembering that a theory is a hypothesis that has been proven over and over and over again, can a theory be disproven? And the answer is yes, it can, but it's not tremendously likely. Disproving a theory would be the exception, as these gentlemen did, as opposed to the rule. So how do we form a hypothesis? We spend a moment of time here thinking about how hypotheses are formed. And so to form a hypothesis, we use what we call inductive reasoning. And so inductive reasoning takes a series of specific observations and molds them together to form this general principle. And so we may look out in the natural world and see that cardinals, if we look at a bird, this bird that we see in central Kentucky, a cardinal, we notice that the males are brightly colored or bright red in color, and the females are drab brown in color. And we might look at another species, so that's one observation. We might look at another species of bird, a wood duck. And when we look at the wood ducks, we see, once again, male, brightly colored, female, dull colored. When we look at yet another species of bird, a mallard, we see male, brightly colored, female, dull colored. So there are three observations. We have these three specific observations, and we can think about that and say, okay, why is it that in all of these cases, the male is brightly colored and the female is not? She's just very dull and drab. And so we may say, okay, what's the best explanation for these different observations? And we can say, in this case, perhaps it is that the males are brightly colored so they can attract and mate. They can attract females. And so that would be an example of inductive reasoning. We have a series of specific observations molded together to form a general principle. So then we'd want to test that hypothesis. How do we test that? To test that, we use something called deductive reasoning. And so deductive reasoning uses general principles to predict an observation. And so let's stick with our example for a second. So we can say if males are brightly colored to attract females, then brightly colored males would be expected to attract more females than dull colored males. And so we can catch a bunch of cardinals and we can dye their feathers. We can make some of them super bright red and others a very dull color of red, or maybe even brown, and release them into the environment and see how many mates they attracted. And so we can form an if-then statement as we perform this hypothesis. We can say if males, the bright color of males are to attract females, then brightly colored males will attract more females than dull colored males, and we can test that very easily. So deductive reasoning is used to take general principles and predict an observation. Inductive reasoning is used to take a bunch of separate observations and form this general principle. At this point, we're going to talk briefly in this chapter about experiments. And so we'll come back to this slide, but we want to start with our experimental method slide. And so the point of doing an experiment is to collect data. And in an experiment, we have at least two variables that are part of the experiment. And so we have an independent variable that is manipulated, and we have a dependent variable that is measured. So the dependent variable is always the one that we are measuring. A good way to think about it, in my opinion, is that the dependent variable depends and is going to change in response to the independent variable. If we look back now at our slide, then we see a an experiment that has been conducted, and we can very easily see the dependent and independent variable in this experiment. So this is from our text, and this is a couple of years old at this point, but a few years ago, the olestra that was incorporated into certain potato chips was a big deal, and it was causing some intestinal difficulties, and a lot of people were talking about it. So in this particular case, the researchers do an experiment, and they test to see, does olestra cause intestinal cramps? And they have an experimental group and a control group, which we'll talk about here in just a moment. But as we focus on the dependent and independent variable here, we see that the dependent variable is, do the subjects of this experiment get cramps? And so we're measuring to see if these people get cramps or not after either consuming olestra or consuming potato chips that do not have olestra. And so the independent variable is olestra. We either give them olestra or we don't. That's what we are manipulating. The independent variable is what you, as a researcher, have control over. You are changing something and then determining what is altered in the subjects of the experiment in response to your changes in the independent variable. So here, the researchers either give the subjects olestra or they don't, and then they measure whether the subject of the experiment get cramps or not. So dependent variable is getting cramps. Independent variable is olestra. Either the subjects get olestra or they don't. Now we also have an experimental. If we go back to this slide, we have the idea of a control group introduced at the bottom here. And so why do we have a control group? That's a question you'll be asked on future quizzes and exams. And we most likely realize the answer to that question at this point. But here in this experiment, we have an experimental group and a control group. The experimental group has the independent variable manipulated. In other words, olestra is added to their potato chips. And in the control group, the independent variable is held constant. Olestra is not added to those potato chips. And so the point of having a control group, as you can see from the experiment, is to have something to compare to. So we want to know what the natural state is, so that when we manipulate the independent variable, we have to know, is the change that we observe a result of the independent variable or the result of something else? And so the control group allows us to address that question. The control group has the independent variable held constant. The independent variable is different in the control group than in the experimental group. So a very interesting aspect of experiments is this idea. We do not fortunately do experiments on humans today. We universally, I would hope, consider that unethical. And so the question becomes, we need to gain this information on how physiology, how bodies work, but we can't do experiments on humans. And so what do? And the answer is that we oftentimes use animals as substitutes for humans in scientific experiments. And there's some controversy surrounding that, but it's a universal practice. It's good today that we have many rules, in fact a tremendous number of rules that govern and what you can and cannot do with animals in an experimental setting. There are these committees called Animal Care and Use Committees that are present at universities that dictate what you can do with animals very specifically and how animals have to be treated. If you want to do an experiment with animals, you have to come up with a very specific plan in order to ensure that the animals are treated humanely. So most commonly in biology today, we use mice as a substitute for humans. Now, there are occasions in which other organisms are used. It's very common to use fruit flies for certain experiments. And sometimes it's more advanced animals are used. And so things like dogs or even monkeys in rare cases are used for experiments. But mice are a great little, what we call a model organism. A model organism is an organism that stands in for humans. We're not going to do experiments on humans, but we're willing to do experiments on a model organism. And so mice we're pretty sophisticated with the mouse research that we do today. we oftentimes, as it says on the bottom point of our slide here, the third bullet, we genetically modify organisms to be more like humans. And so we can take a mouse and we can place a human immune system into that mouse, or at least a portion of the human immune system, or we can make part of the heart cells of that mouse human, and that makes that mouse much more valuable as a research tool than just the mouse that you would find running around in your basement. If we look at the screen, we see on the bottom left some genetically modified mice. These are glowing mice, mice that have the green fluorescent protein inserted. So you see that 2 on the far left and 1 on the right have the GFP gene inserted, and the 3 in the middle do not. Interestingly, if we look at the right-hand side, we see another genetic modification. In this particular case, the mouse on the left has had the gene leptin, L-E-P-T-I-N, removed from its genome. And so leptin is a satiety factor. It makes us feel full. All of us possess leptin. It is a gene that's primarily expressed in adipose cells and fat cells. And so the more those we have, the more full we feel. The mouse on the left has had that gene removed, and so it never feels full. And so it eats and eats and eats until it obtains this massive size that you see there. Its sides are dragging along the ground. And of course the mouse to the right is a normal mouse, what we call a wild type mouse, and so it has the normal leptin gene. And so this demonstrates pretty conclusively what the role that leptin plays in mouse physiology and presumably in human physiology as well. That's many people are working on that research question today. What role does leptin play in human health, and could leptin be manipulated to make some sort of appetite suppressant that would be perfectly safe for humans to consume? As we finish our discussion of experiments, we want to talk briefly about correlations. So correlations are relationships between 2 variables that we observe in an experimental setting. And so on the screen, we have a good example of that. We have on the x-axis, the x-axis, A-X-I-S, axis, is the horizontal axis, the y-axis is the vertical axis. And so on the x-axis, we have a psychological stress index. And so the more stressed a person is, the further to the right they are on that psychological stress index. On the y-axis, we have the percentage of subjects that have colds, and so 25% at the bottom and 50% at the top. And so what we see is this correlation between psychological stress index and the number of students that have colds. So the more stressed these people are, the more colds that they have. And so that's a correlation. We have a distinct correlation between number of subjects with colds and psychological stress index. The question becomes, is the stress that the person is feeling causing the colds? And so a correlation can be observed. And then we have to ask the question. We don't want to automatically assume that one variable is causing the other. We have to ask the question, is there a causative sort of relationship between the 2 variables? So it seems that it could be the case, right? High stress, as we see in part A on the top of our figure here, causes a high cold frequency. But what we need to consider is this, that sometimes other things could be contributing to both of the variables that appear to be correlated. For instance, the person could have another illness of some type, and that contributes both to, as we look at the bottom part of our figure here, the other illness contributes to the high stress, and it also contributes to the high cold frequency. Or perhaps the person's eating a poor diet. They're a college student and they aren't taking time to eat meals, nutritious meals, and so that contributes to stress, and that also contributes to high cold frequency. Little exercise may cause stress and a high cold frequency as well, and little sleep, perhaps. Lack of sleep might be the best factor of all 4 of these. Little lack of sleep may cause high stress, and it would also certainly make you more susceptible to a cold. And so the point of bringing this up is to point out that just because 2 variables are correlated doesn't mean that one causes the other. And so in science we have to be very careful when we observe a correlation. We might be very excited at first, but it's important for us to step back and say, okay, Even though there's a correlation, is there a causative relationship between these two variables? We close our discussion of experiments by talking about statistics. And so statistics are a branch of math that allows us to determine if an event that we observe in an experiment is a result of random chance or an actual result of the independent variable. And so statistics allow us to determine whether what we're seeing is actually meaningful or not. Statistics also tell us, based on what I just pointed out there, if what we're seeing is truly meaningful, then we can expand the results of our experiment potentially to the entire population. So if we get a valid result, we can say, okay, this is perhaps valuable for all humans, not just the humans that are part of our little experiment here. So here's an example of how we use statistics. So we have an experiment here shown in this graph on the right-hand side of our figure, and on the y-axis we have the average number of days that it takes a person to get over a cold. On the x-axis we have two different categories. On the left-hand category is people that took a placebo, and we'll explain what that is in a moment. On the right hand, we have a group of people that took zinc lozenges. And so the question is, does zinc help you recover from a cold more quickly? So to address that question, we did the following. A number of people were intentionally infected with a cold. Now these are people that are well compensated for their participation in the experiment. And of course it's 100% voluntary, but they wanted to make some money and be part of this experiment. So the people that are given a cold are divided into two groups. Half of the people get a placebo, which is just a sugar pill. It doesn't have any of the independent variable in it. The independent variable in this case, as you realize, is zinc. So the placebo is given so that the people will not know that they are not receiving the actual treatment, the actual independent variable. That's important because of this thing we call the placebo effect. When you think you're going to get better, sometimes you do, and there are simple physiological explanations for that. Things like the release of cortisol, which is the stress hormone. If you think you're taking a pill that's going to make you better, you'll be less stressed as a result of less release of cortisol. Your immune system will be less suppressed and you get better. So anyway, back to this idea. The people that half the group takes a placebo and half the group takes zinc lozenges. So now it looks initially like the zinc's having an effect because it took these people, sorry this axis is curved over a little bit, but it took people that took the placebo about 7.5 days on average to get over a cold. It took the group of people that took zinc about 4.5 days to get over the cold. But the question is, is this meaningful? Are the results that we see, do they actually mean anything? And in order to determine that, we have to apply statistics to this particular experiment. On the left-hand side, we see the what we refer to as confidence intervals that are extremely small. The little red bar here that I'm pointing to with the cursor here and here represents what we call a confidence interval. And so in very simple terms, here's what a confidence interval means. Now we think that the average time that it took the control group to get over the cold was about 8.5 days. That was the average time as calculated based on our data. But when we use statistics tell us that real number is between, what would that be, 7.5 and 9. So the calculated number is 8.5, but statistics tell us that there is a 95% chance, this number over here, there's a 95% chance, because this is a 95% confidence interval, 95% chance that the number is between 9 and 7.5. So we're certain, at least 95% certain, that the number is somewhere in this red area here. The same thing applies to the experimental group. So ask yourself this question: are these two numbers different? Are they definitely different? And you'll have to say yes, they are, because the lowest the top number can be is 7.5. The highest this number can be is about 5, I believe. And so there's no overlap. The key is there's no overlap. Sorry about that. There's no overlap between the top of this confidence interval and the bottom of that one. On the other hand, if we have high confidence intervals, then we do have overlap. And so this means that these two numbers, remember this is a different experiment here, and so we came up with different confidence intervals based on our statistical calculations. The means are the same, as you can see from the left-hand group over here to the right-hand group, but the confidence intervals are different. There is overlap here, and so these two means, we cannot say with 95% confidence that they are different, because it could be that this lower number is up here, the higher number is down here, and so they're actually the same. There is no difference between them. So that is the significance of statistics in scientific studies. Statistics tell us how meaningful our results are. Are our results the product of random chance, or are they the result of the manipulation of the independent variable? We close our discussion here in Chapter 1 by talking about sources by which we obtain scientific information. All scientific information ultimately comes from primary sources. So primary sources are research articles that are published by scientists. Now, a key idea here is this, that all primary sources are peer-reviewed. So we see that right here as I'm underlining with the cursor on our slide. Peer review means this: I write a research paper, I send it off to be reviewed by other scientists. The other scientists review it. They say, yes, this is a great paper. No, this is this paper is a piece of junk. And so if it is positively reviewed, then that paper will go into a scientific journal. If it's negatively reviewed, it does not go into that journal. And this is how all science works. 100% of science today works in this particular manner. Scientists do experiments, they submit their experiments for publication, and if the publications are accepted, then those results go into these databases that are all online today, as you undoubtedly realize, as the bottom bullet says here. And anyone in the world can access those. In fact, today I saw that just a few minutes ago, I saw that a journal article that I published a few years ago was cited by some people doing work in China. It's not an especially significant journal article that I published, but a couple people have found it in different useful in work that they're doing. And so it's very much universally available around the world today as we look at these databases. The other place that we can get scientific information is this, from secondary sources. And so secondary sources include books, internet, news reports, advertisements, and secondary sources are sources in which they go to someone else goes to the primary literature and pulls out a bunch of different primary sources regarding a topic, compiles those, and then presents it together. And so this is a more common way to get scientific information for a non-scientist. Few non-scientists are reading primary literature, but many people are reading news reports and books, and especially internet articles regarding primary literature. Now remember, secondary sources are not as accurate, not as trustworthy as primary sources, because of course it would be easy to go to the primary literature and pull out, let's say that there's a particular topic and there have been 1,000 studies done, and 995 of those studies have found one result, and 5 studies have found a different result. And so you could go to the primary literature and pull out those 5 studies. Remember, there are only a tiny percentage, only 5 out of 1,000 studies that get this result. You could write an internet article and you could say, this is how it is, look at these scientific studies. But in fact, you'd be wrong, because at least your results would be very suspect, because most of the primary literature has found the opposite conclusion of what you're presenting. So you have to be careful when you look at secondary sources and view them from it with a critical mindset. Hopefully they're perfectly honest and have done everything above board, but it's possible that the people that are presenting the information have an agenda. Most likely the agenda is going to be they're trying to sell you something via an advertisement. So you have to consider the source of those media reports. And that's what I say on this slide as well. So anything can be posted on the internet, and you have to be extremely careful about what you find there. And there's all kinds of pseudoscientific or non-scientific products that are pushed in today's world. And so if you look at the Nature's Way, this all-natural sort of seed extract there. And so interestingly, these are products that are non-FDA approved. Something that's not FDA approved doesn't have to go through the stringent testing that drugs and other more reliable sorts of health supplements have to go through. So you have to be careful with that. It's not illegal to sell you a sugar pill, as long as it doesn't hurt you. It's not illegal to sell you something that doesn't work. And so many times these sorts of products will try to twist scientific information to talk you into buying something. The final final source through which you may obtain scientific information is from anecdotal evidence. Anecdotal evidence is one person's experience. One person's experience. It's not based on data at all. But for instance, anecdotal evidence would be Shaquille O'Neal doing the advertisement telling you that Icy Hot is the best thing in the world for curing, muscle pain, something like that. Sore muscles, Icy Hot's a great thing. And so, what's that worth? Well, I mean, we have obviously serious doubts that he even means that, since he's receiving a huge amount of money for presenting that information to us. But even if he did mean it, it wouldn't mean all that much. It'd be a place to start, perhaps, in the scientific method. But since anecdotal evidence is one person's experience, then we have to view it with a great deal of caution. There are all sorts of factors that can lead to a result, and since we're looking at a sample size of one, anecdotal evidence is one person's experience, then we want to be very cautious as we assign any sort of value to anecdotal evidence. This concludes our discussion of the scientific method in Chapter 1.